Hi, everyone. My name is Pranam. I'm excited to be here. I'm a professor at Penn in bioengineering and computer science. Today, I'm going to share with you a few stories from my lab on where we're advancing genetic agency, but by developing programmable biologics. And so I always like to start with my lab. The lab is a very interesting setup where we have a full computational team, pure AI scientists developing generative models that design protein therapeutics that my experimental team can go then take and translate. But we develop, and we operate on a very large spectrum, all the way from theoretical frameworks all the way to in vivo disease models, and can get molecules into preclinical studies quickly. So we develop programmable proteins that can increase our genetic agency. I'm going to show you a few examples. For example, we develop proteins that can edit DNA sequences. In the last few years, we've developed models like SPAMALOT, which are sequence-based models that design broad targeting CRISPR enzymes that give us broad DNA targeting capacities, allowing us to edit mutations that were previously inaccessible. Now, we also develop proteins that can generate new cells and new tissues. We've developed graph theory algorithms that can identify transcription factor proteins, that if you put these proteins into stem cells, now we can convert these stem cells into ovarian support cells. And working with my company Gameto, we've then taken these ovarian support cells, we co-culture them with immature eggs that allows us to fertilize them into mature eggs. And now we have multiple babies born from this technology. Actually, Gameto is the first US company to achieve a phase 3 with a stem cell product. So it just goes to show you that I think that this idea of a functional algorithm to a protein therapeutic can actually work. But what we're really focused on now is developing these biologics that are able to bind, and not just bind, but also modulate other proteins. And because look, we think genes are very important, but it's really at the end of the day a lot of proteins that cause disease. And if you look at over 300 of the intractable cancer-causing proteins, a lot of them are just undruggable. They don't have binding pockets, so you're stuck there. And moreover, a lot of them look like this. They look like spaghetti. So it's not going to make sense to use structure-based methods here to model these proteins and design molecules for them. And so the question we've asked is, can we do the next best thing? Instead of small molecules, can we make drug-like peptides to bind to these proteins? And then can we use them to degrade these undruggable targets without needing any structural input? Because in our first instinct there was to use protein language models. Obviously, you guys know these. They've been trained on millions of protein sequences to capture properties of these proteins from structure, function, and interaction. We can take these protein language models and we can perturb their representations to generate new peptides that potentially could be useful. But then we take those peptide sequences that we generate and we throw them through a separate language model that we train to learn peptide protein targeting specificity, a contrastive CLIP-like model. And so this peptide prioritization with CLIP, or PepPrCLIP as we call it, works really well. It's just a sequence-based model, but as you can see, these PepPrCLIP peptides bind with nanomolar affinity to their targets. If we use these peptides and we use them to inhibit enzymes that have specific activities, you can see that on the left, PepPrCLIP peptides do a really great job at binding and inhibiting enzyme activity versus structure-based methods on structured proteins. We have this really cool technology that for those of you who are interested in genetic therapies, we take these peptides and in the lab we attach them to the catalytic domain of PTM enzymes like E3 ligases. This allows us to not only bind, but degrade proteins. This is beta-catenin, a core cancer driver. You see the bar goes down because we're getting rid of these proteins, and we also result in reduction in Wnt signaling in those cells as well. So not just binding and degrading, but also functional activity. But then this is where proteins that you really cannot use structure, this is where the type of proteins we're talking about, SS18, SSX1, completely disordered, drives synovial sarcoma. We found a peptide just by testing 10 that was able to bind to this protein, and in synovial sarcoma models, we're able to completely ablate this completely undruggable, heavily disordered protein. This is not something a structure-based method could ever do. And so you can also do this conditionally, right? So we've fine-tuned protein language models to say, hey, if you give me a new target sequence, just the sequence, and you tell me, hey, I want a binder of length 5 to that protein, PepMLM that we've trained can unmask and generate that binder. It almost sounds kind of silly and stupid, but it works. Actually, PepMLM on structured targets achieves a hit rate of over 40%, so you only really need to test a few to get a good binder, as compared to structure-based methods on structured targets having a lower hit rate. So we said, let's go to these really hard targets that maybe you guys in the audience are interested in. Huntington's disease. It's caused by this mutant Huntington protein that degenerates neurons. We just tested 5 from the sequence model, and as you can see, all of them with our Huntington collaborators showed complete removal of Huntington protein. We can go into viral proteins and very disordered phosphoproteins, and you can see this is the viral proteins on the left, in green, and when we treat them with these peptides, we're able to completely remove these proteins. But that's great if a protein is bad. What if you want to keep a good protein? For example, p53 is ubiquitinated by its E3 ligase MDM2, and for a lot of diseases, you want to keep wild-type p53 around. So could we stabilize these proteins? The answer is yes. We say, let's take our peptide, and instead of an E3 ligase, let's attach it to a deubiquitinase, and that allows us to stabilize proteins. And as you can see now, the bars go up, because no longer are we're removing the ubiquitin chains from these targets. We can take these deubiquibodies, and in the lab, we could put them in an AAV, but we do a lot of LNP work. We'll put these as mRNA in an LNP. We deliver these, and as you can see, not only do we upregulate or destabilize proteins like p53, but that also induces the apoptosis in these tumor cell models. Can we target other things? It doesn't matter, but proteins are great, but what about molecules like pollutants, like PFAS or metals? Take metals, for example. They pollute our environment, and they also create a lot of health risks. So you may be thinking this is not a very good therapeutic target, but in reality, if you were able to sequester these heavy metals, we could reduce these toxicities. So we've developed models like Metal Laureate, which essentially takes a model like Metal Latte that we trained to learn the interaction between proteins and metals. You can see it's pretty accurate across a lot of heavy metals. And we can apply both diffusion on the protein representations, but also on the label of which metal it binds to, such that in the reverse process, we not only regenerate the protein sequence, but we can make sure that it binds to a very specific heavy metal. And we've developed really interesting metal binding assays to make sure that these binders bind to metals like copper, to zinc. We're actually putting these peptides for nickel in cows to reduce methane production because they use nickel as a substrate. But even if we can get more specific to these targets, it's just not enough to bind to them. Binding is just very simple and not very important. What matters is they need to become therapeutics, right? And what do I mean by that? Therapeutic design is multi-objective, right? It's not just about binding to your target. It's about being soluble, being orally bioavailable, having a long half-life, not sticking to random things, and also not being toxic. And you guys could throw out another set of properties you want in your therapeutic. And so to fix that, we've developed models like PepTune, which can do multi-objective guided generation of therapeutic peptides, but it works for other molecules as well. The idea is that we use chemical language to represent peptides, so we can model side chain stereochemistries, non-canonical amino acids, and others. That is really nice because we use this type of architecture called mass discrete diffusion to generate new peptides. As you can see, mDLMs, you can get cyclizations, non-canonical amino acids, you can get very diverse peptides, which are nice. But now we innovate a new method called Monte Carlo tree guidance, which allows us to not only generate peptides, score them with different property predictors, but actually adjust the generative model to only produce sequences that produce these Pareto-optimal peptide sequences. And as you can see, we can make binders that bind to 2 targets, that are soluble, are not toxic, don't have non-fouling properties. And we have been able to create new GLP-1R agonists this way, antagonist peptides for receptors like AMHR2, and to create dual peptide binders to bind to targets like transferrin and GLAS so that we can recruit 2 proteins together. The PepTune framework is super versatile. I'm just going to show you few last things. We've used PepTune to do mRNA optimization. We even have a company doing this where we can optimize for half-life, ribosome load, translation rate, and we've shown that these optimized mRNAs are not only super stable, but they also have better activity as well and translation capacity. And finally, we are also making better binders that are isoform-specific, mutant-specific for variant effects, as well as for PTMs as well. So models like SOPIA help us happen. So the last thing I'll say is that the future of genetic agency, I think, will bridge theory that develops these new algorithms, new experimental modalities that we still really need. We can't just be delivering a gene forever, as well as good therapeutic translation and fast if we can do it. So, and that's exactly what we're doing with the models we're developing. So with that, thank you. I know we went through a lot, but I really want to thank my lab. They're the best. I'm so proud of them. I'm just their weird spokesperson. We have great funding, thankfully, that's sticking around for us, and great collaborators. If you're interested in any of this work, even if you're a company, we'd love to work with you and give you molecules, whatever you need. And thank you for listening to our stories. And if you want to follow our work, we're a pretty young lab, just moved to Penn from Duke. And if you want to use our models, they're all on Hugging Face, and they're all open sourced. So feel free and to have at it. Thank you so much.
Thank you, Pranam. Thanks so much.